Good afternoon, students. My name is Harender and I am from Department of Biological Sciences and Bioengineering from IIT Kanpur. So we are trying to understand the principles and processes of uh, a part of biology, that is biotechnology, which is chapter 9 of class 12. So before entering into today's session, let's have a brief recap on yesterday's session, previous session. So that we'll have a continuity in our understanding. So in the first part, we were like mostly learning about what are the basic principles of the biotechnology and what are the important tools of it. And in the yesterday's session, we were mostly discussing about the different processes that are involved in biotechnology. So we were discussing about the process involved in biotechnology. And in this process, yesterday we learned about isolation of DNA. Now, why do we need to isolate our DNA? Why? Why do we need to isolate? Because it contains our gene of interest, right? And I was like like I commonly take the example of insulin because we know that many of our relatives or like someone we know is taking insulin shots. So we are taking the protein, that is insulin protein, which has its function. So always remember, a gene will form, which is a DNA, will give rise to mRNA, and mRNA will give rise to protein. And this protein will have different functions. Like if it's a function, it will be called as a functional protein. Functional protein. Then there are structural proteins, globular proteins, etc. So at the end of the day, like at the end of the central dogma of the process, it's the protein that has the activity. For example, when I'm telling you about an enzyme that is EcoR1, BAMH1, HINT3, anything, any enzyme, tetracycline resistance, ampicillin resistance, everything has its function. And this function is not because of the gene, it is because of the gene, obviously. But at the foot level, at the foot soldier level, the proteins are the ones that are doing it, right? Now, so we understand that why do we need to isolate the DNA? It is because it will contain a gene of interest, and this gene of interest will give rise to the protein. The foot soldiers, basically, they are the foot soldiers. They will take up whatever we want. Now, if this gene of interest can be present in either bacteria or, for example, all the restriction enzymes, they are isolated from bacteria. Then they can either be from animal cells or they can also be from plants. Right. Now, we yesterday understood that this bacteria or the plants have very rigid and very strong cell wall. Right? Another example that we read yesterday was fungus. So, for example, the fungus cell wall, fungus cell wall is made up of chitin. The color, the fungus cell wall is made up of chitin. And in order to degrade this, like in order to destroy this, like break this chitin, it is called chitinase. The enzyme used here was chitinase. And this chitinase was able to degrade the chitin so that we can easily isolate the DNA inside the fungus. Moving on, we have plants, and this plant cell wall is made up of again one of the like very special material, and I think you might have like heard it often as well. It is made up of cellulose, cellulose. And to degrade or to destroy or to break this cellulose, we will use another enzyme called cellulase. The difference is here there is O, but here we are using ACE. Then animal does not have any cell wall, it just has one plasma membrane, so it is fine. And the bacterial cell wall is made up of peptidoglycan. This is again one of the special polymer, peptidoglycan. And to break that, we'll use lysozyme. Lysozyme. So this again is an enzyme. So we can understand that how it is very we can understand it as a very necessary thing. For example, that this bacteria, like every cell has this plasma membrane, right? Every cell has this plasma membrane. But depending on the cell type, depending on the origin of the cell, we'll have a extra layer as well. For example, this is a normal animal cell, right? Animal cell. But if it has a separate extra membrane that is which is made up of cellulose, then it is a plant cell. And if the extra membrane is made up of chitin, chitin, then it is called it is a fungus. If it is made up of peptidoglycan, peptidoglycan, it will be of it will be of bacteria. So every cell, every organism has this plasma membrane common, but there is a special coating, a special covering of this cell, which is very important. It can be, and if it is made up of peptidoglycan, it is of bacterial. If it is made up of cellulose, it is plant, and if it is chitin, it is fungus. So we use this enzymes to degrade that material. So what we learned here, that for the bacteria, cell wall type. So what I'm summarizing here, bacteria, then we have fungi, then we have animals, and we have plant. So the bacterial cell wall is made up of peptidoglycan. Fungus is made up of chitin. Animal, it does not have cell wall. Plant, it is made up of cellulose. Now to degrade this, we need to use enzymes such as lysozyme. So you must remember that in your home people use like Lysol, etc. So like just use that terminology and remember that when this various cleaners commercial comes up, they they basically confirm that they kill a lot of bacteria. So like for example Harpic, and then one is Lysol. So from Lysol, just remember there is a lysozyme, and this lysozyme can kill bacteria, basically break off the bacteria. Then for chitin, we have chitinase. Chitinase. Since animal does not have it, not will break upon the shear force. And for cellulose, we will have cellulase. So just one difference: cellulose. Here there is O, and here it is A. So this is it. Now once this cell wall breaks, then all the things that is present inside the cell comes out, such as DNA, obviously. Then there might be RNA, and then there will be protein, and other things as well, like such as polysaccharides, etc. So we won't go into that deep. Now, once they break open, what we do next is we'll have to remove all this contaminant, because our desired product is DNA and rest of it is a contaminant. So for RNA removal, we use RNase, and for protein, we use proteases. Protein. So I hope you remember this enzyme names, because they are very important from the any exam point of view. So you should remember and memorize every single of them. Peptidoglycan will be degraded by lysozyme, chitin by chitinase, cellulose by cellulase. So you just for in term in case of fungi and plant, it is just a common correlation. So you can easily remember that, I guess, because it is chitin and it is chitin. So whenever there is an enzyme, there is ase or chiases. As you can see RNase is an enzyme which can make RNA, protease can protein, and so something like that. Now, once DNA is isolated, what we further move up was once the DNA is isolated, now we have the DNA. Now we have to fragment the DNA, because the DNA is very big. And let's say our DNA synthesis here, then we just have to cut this DNA. And for that, we use molecular obviously, we know that. But in order to separate this small fragment from the rest of them, we use a special technique called agarose gel electrophoresis. Now, in agarose gel electrophoresis, we use a special polymer, that is agarose, obtained from seaweed and algae. And this is the property thing you should remember this again. It has a sieving property, and sieving property basically defines that it can separate DNA based on size and charge. So now we know that this is agarose gel, and this is where we loaded our samples. Now after digestion, we will have 100 base pair and 1000 base pair DNA basically. Let's suggest that. Now this is negative and this is positive, and we know that DNA is negatively charged, so it should move towards the anode. DNA move towards anode. So once we load it after digestion, the sieving is basically a property of being porous, has the pores. So the smaller the smaller one will easily pass, and then it will come here 100 base pair, and the bigger one won't, will pass very slowly, and therefore we'll have a difference like this. And this is how it is based on the size and the charge. Is some network issues. So what I was teaching here was, so now we have already loaded our after the DNA was digested. We did agarose gel electrophoresis. Right. So this DNA bands are cut out from the agarose gel and this is called elution. DNA bands are cut out from the agarose gel and this is called elution. So once we run the DNA, okay, let me finish this slide and then we will discuss. So the DNA bands are cut out from the agarose gel and this step is called elution. Okay. Now the cutout gene of interest from the source DNA and cutout vector are mixed and IGS is added. It creates a recombinant DNA. So what they're trying to say is, okay, so this is the gel and this is our ladder. What is ladder? Ladder is a solution of concentrated DNA strands having a definitive size. So, like for example, let's say this is a ladder, my ladder vial. I'm like ladder tube. I have some liquid. Now this liquid will have DNA of variable length. So for example, if I load it on gel based on the size, it will separate. So let's say like this. So let's say this is 50 base pair and 10 kb. So this is how, and then this will be 100 base pair. This will be 50 base pair. This is 5 base pair. Then 1 kb, 1.5 kb, 3 kb, 5 kb, 7 kb, 8 kb, and something like this. So this is how it will separate. So this solution, this mixture will have all the DNA and based on the size, based on the RNAs, sorry, and the agarose gel, they will be separated very easily. Then after this, after loading the ladder, I'll load undigested undigested DNA. So like let's say my DNA is of 2 kb, and since I didn't digest it, and if I load it and if my gel runs, then it will be just coming just exactly at 2 kb. So here it's a 2 kb, right? kb. Now I digested my DNA with the containing the gene of interest with let's say eco R1 and hint3, and I know that once I digest, I'll get a product of let's say this very big, and this is my gene of interest. So if I cut here, I'll get a product of 100 base pair. Now, my size is 2.1, that is 2100 base pair. And if I cut it, I'll get two bands basically. One will be of 2 kb, 2000 base pair, and one will be 1100 base pair, right? That is how it is to 1100. So I'll see that I'll get one band here, this 2000 base pair, and one will be here. And this is what my gene of interest is. Gene of interest. Now, I just want this. So what I'll do is, I'll basically cut this gel, small it, and I'll put it in a tube. This gel has my gene of interest, and basically I'll heat it at 50 degree and melt, and the DNA again will be in the solution. Melt. After this, I'll purify it. Basically, I will remove all the agarose and all the like whatever the buffer was used. And once I purify it, I'll get my pure gene of interest. So I hope you have understood. And this process of cutting and extracting the and purifying or extracting the gene of interest is called elution. Elution is process by which the DNA cut from gel and purified. So this is what the entire elution is. So after digestion, I'll load it on my gel and I'll see my separation of gene of interest is 100 whisker. I already know that, and I'll cut the gel here and I'll melt it basically and then purify the gene of interest from there. And this whole process is called elution. And so elution is there. Now similarly, we'll do the same thing with the vector DNA, vector, and then we'll do the ligation. So that was with the gene of interest. Again, we have this vector. We know that we want to put the insulin gene in a bacterial vector. So we'll cut it same thing with the same enzymes, equal and hint3, and we'll run it on gel. And this time we want this backbone, which is on 7 kb, and this is the extra piece that we cut so that we can introduce our side here. So we'll get 7 kb here. We'll cut again this gel and purify, elute it, and we are ready with both insert and sample. And once we obtain both insert and sample after digestion and after purification, we'll use ligase for them to join to ligate. So what happens here? That we have purified our gene of interest, we have purified our vector, and we added ligase. And once we add ligase and incubate them, some of this thing will join and they will form like this. And this is called recombinant DNA. So that is the whole process here. As you can see here, that the band was excised from this, again added here, then at 65, so step for 35, 60, 65 step for 10 minutes, and then it was basically washed and purified, and the DNA was eluted. And then we'll have this gene of interest, and then vector DNA will add ligase, and we'll get a recombinant DNA. Now, now we got this recombinant DNA, right? We got this recombinant DNA. So now we have our recombinant DNA, which has our gene of interest. Gene of interest. This is our recombinant DNA, and then we basically transform into the cell, bacterial cell mostly. Now, if this, and then if you want more of this, we'll basically culture this bacteria in a broth. Culture in broth. And as this bacteria divide, will and again if we lyse this cell, will isolate the plasmid, and our gene of interest is amplified. So this again, the more the bacteria divides, the more the higher the copy number, and everything, the higher will be the gene of interest, right? And since this amplification of the gene of interest uses a cell, this is cell-based amplification, which is uh, time-consuming, a cost, very costly as well, because we need a lot of kits and everything. Yeah, this cell-based amplification. So this is very costly, time-consuming, because it takes one complete day. Like basically, what I will do is basically after transformation, you will get colonies, and then colonies you will basically inoculate again in the nutrient broth, and very next day you will come and isolate again the plasmid. So it is very time consuming, costly and everything. So alternative to this amplification method, we have another amplification, and that is using PCR, that is polymerase chain reaction. So this, first of all, you should know one thing: this is the cell-free amplification of DNA. Polymerase chain reaction is synthesis of multiple copies of the gene of interest in vitro using two sets of primers and the enzyme DNA polymerase. So again, please follow me through thoroughly. The polymerase chain reaction is the synthesis of multiple copies of gene of interest in vitro, multiple copies of the gene of interest in vitro using two sets of primers and the enzyme DNA polymerase. So this is a cell-free because there is no cells here. And again, this machine is the one that is used to do PCR, and this machine is called thermal cycler. This thermal cycler. Now, with this process, we can get a multiple copies of the gene of interest in vitro using two sets of primers and the enzyme DNA polymerase. So here, this is a very important technique, and for this technique, so let me write the name of the technique is PCR, is polymerase chain reaction. So do you guys know, like, I mean, was an accidental, like not accidental discovery. It was developed by one of the great scientists, that is Carey Mullis. And this guy was like, once he was just traveling, and then that's how he came across an idea that if the cell machinery uses all the DNA polymerase and everything that is already present in the cell to amplify and replicate that DNA, why can't we use those enzymes directly and provide an optimum condition to generate such fragments? And this Carey Mullis was awarded Nobel Prize for this. Nobel Prize for this, because it was like one of the best discovery ever. As I said, the cell-based amplification, that uses bacteria to amplify a gene of interest, is very tedious, very consuming, time-consuming, and very costly. But this PCR reaction using the instrument used is called thermal cycler. Uses very limited resources to amplify the gene of interest, and that to many folds. That's not even possible by a normal medi or maximum. So this thermal cycle. Now, you might have studied about replication in class 11th, I guess, where there is the use of DNA polymerase. Then we get a lot of DNTP, and we have this. This is the enzyme. We have DNTP. Yeah, these three things, and then we have primers. So even the PCR uses the same thing. It will have a DNA polymerase, and the DNA polymerase that is used here called Taq polymerase. It is isolated from Hermes aquaticus. And then we have this DNTP. DNTPs are basically what? ATPC. So as the strand needs to elongate, this DNA polymerase will take up this DNTPs and use it. So this is nitrogen based, like nucleotide triphosphates. And then we have primers. Primers are like again very important topic. What are they? Primers are basically small chemically synthesized oligonucleotides that are complementary to the region of the DNA. Now, the primers are oligonucleotides. Short primers, short oligonucleotide, short oligonucleotide, which is complementary to our gene of interest or DNA. Now let me tell you. Now let's say this is our gene of interest. This is our DNA, and in this DNA, our gene of interest is here. Now to just amplify this gene of interest, I want I will design a specific short oligonucleotide, synthetically designed short oligonucleotide which is complementary to this region. So I'll have this forward primer and reverse primer. And once I give this primer, they will go, since they're complementary to each other. Like for example, if my DNA strand has A, T, G, C, I'll design a primer which will have T, A, C, G. So it will complement, it has this complementarity and it will go and automatically bind to it. Right? It will automatically go and bind to it. So that's how the both forward primer and reverse primer will bind. Then there is the action of DNA and polymerase take this. It will start adding the DNTP that is present in the cells, in the mixture reaction mixture, and this NTP DNTP will be used up, and then the chain will be made. So that's the thing. For PCR, we just need template, that is our DNA. We need primers. We need tag balls. So we have understood that polymerase chain reaction of PCR is the synthesis of multiple copies of the gene of interest in vitro using two sets of primers. So we use only one pair of primers and then DNA polymerase. So primers, DNA polymerase, DNTP are the ones that are used in the PCR reaction. Now, primers are basically small chemically synthesized oligonucleotides that are complementary to the region of the DNA. So there are three steps of PCR. Three steps of PCR. First is denaturation, second is annealing, and third is extension. So what are the steps of PCR? First is denaturation, second is annealing, and third is extension. Now you can see here that there's this denaturation. Now we know that the DNA is double stranded. As you can see here, for your representation, or normally like for you to understand, often in this textbook, the DNA is like given like this in the straight ladder kind of thing, but it is always spherically like helical, always helical, right? So this is the DNA, and it is double stranded. Now we know that the primer should go and bind to the DNA, but again, if they're already paired up, where will the primer go and bind? So for that, we will need we'll have this process. We will have this step called denaturation. In this step, what happens is we raise the temperature to 95 to 98 degrees, 94 to 98 degrees. What we do here is that we raise the temperature from 94 degrees to 98 degrees, and this leads to the breaking of all hydrogen bonds. So this all the base pairs are joined together, or they are bound together by a hydrogen bond. So once I increase the temperature from 94 to like between 94 to 98, around that, all the hydrogen bonds will break and they will separate. And once they separate, we have this annealing step wherein the primers will go and bind to the complementary DNA. For example, here the bonding was TAA, so here it will be AT, and the rest of them will be not like the rest of them will be like. So as you can see, this cannot bind this primer which has ATT cannot bind anywhere except this region. Therefore, this is how the specificity is. You will design your primer such a way that it will go and bind to that only, to its complementary side only. So that step is called annealing. Annealing will lower the temperature, which leads to binding of the DNA of the primer. Sorry. The first step is denaturation, wherein you will basically break the DNA strand, break all the hydrogen bonds in this, and that will lead to the separation of the DNA strand. And once this DNA separates, we'll lower the temperature, leading to the binding of the primer to the specific region of the DNA. Next, the DNA polymerase and the DNTPs will be used up in order so that there is an extension. So as you can see, like something like this will happen. The DNA polymerase will act like this, and it will keep on adding the base. It will keep on adding, it will keep on taking the base pair, that is deoxynucleotide, from the solution, and it will keep joining it. And that is this process called extension, because initially it was just like this, but then it was extending, and this step is called extension. So denaturation is basically breaking of the DNA, and annealing is basically and attaching, or like basically annealing of the primer to the DNA strand. And then extension is by DNA polymerase and DNTPs, where the DNA polymerase uses the DNTPs present in the solution to make the stand, make a new stand. And this is one cycle. And the many times you repeat the cycle, the higher will be the uplift volume. So this is what this. So the first step. So that was the entire overview of what the entire PCR is. So we'll just again we'll go through this again. That step one is denaturation. Denaturation. It is a heating of the target DNA, that is gene of interest, at high temperature, that is 94 degrees, to separate the stand. So from this is how the DNA is. And once we increase the temperature to 94 to 98, we'll get a separate stand. So this is denaturation. So what is denaturation? It is the first step in PCR, that is polymerase chain reaction, wherein by increasing the temperature up to 94 to 98 degree, we basically break the DNA strand and separate them. Now each strand acts as a template for the DNA synthesis. Now each strand can act as a template, because we have two primers. We have one forward primer and one reverse primer. So that we can get a whole double-stranded DNA in the late. So what I was trying to say is that after this DNA strands are separated, there is next step is annealing step. In this annealing step, it is the joining of the two primers that too at like around 52 to 55 degree at the 3' end of the DNA template. As you can see, both are joining at the 3' end. The primers are joining at the 3' end. So why do we need two primers? Because as you can see, we will design like we have this DNA and this is separated DNA, and our gene again will be a double-stranded strand. It will be double-stranded DNA, right? Now we'll design two primers. One will basically link up to the upper one, that is from this reverse primer, and then here again from the 3' end, that will be forward primer. And once they do this amplification, they will be able to amplify this region particularly. After the 30 cycle, 50 cycle, everything, we'll have this region, and that too in this form. Therefore, in order to obtain this specificity, basically, we always use two primers, and yeah, and this double strand. At the end of this process, we'll get a double-stranded gene of this. Now, the second step that we are learning is the annealing step, wherein after the temperature, the lowering of the temperature from 94 to 52, the primers go and bind to the complementary site of the parental DNA, and that is the annealing step. Once this primer binds, at 3' end, by the action of DNA polymerase and deoxin DNTPs, there is a extension step coming up. Now, extension is the third step, that is extension. It is the addition of nucleotides to the primer using a thermostable DNA polymerase called Taq polymerase. Again, this is one of the important questions here. Let me highlight it for you. So again, this is again one of the very important questions here. That in the addition of the nucleotide to the primer, we have added template, we have added primer. Then we'll add another polymerase, a DNA polymerase, which is responsible for the synthesis of the new strand. That is DNA polymerase, which is Taq polymerase. And this Taq polymerase is isolated from Thermus aquaticus, a bacteria. Now, why do we really need this? So, it because unlike normal DNA polymerases that is present naturally in our body or in any bacteria or anything, they can easily degrade at 90. But we need this temperature to break. We need 98 degrees for the this thing to break, for the DNA strands to break. Therefore, but at 90 degrees, the Taq polymerase won't be stable, like, viable. It will basically degrade, right? So therefore, uh, we have isolated up this enzyme from from the bacteria which survives in the high temperature, and that is called Taq polymerase. So the bacteria name is Thermus aquaticus, and from this Thermus aquaticus, we have isolated the enzyme that is Taq polymerase. Now, the specialty of this enzyme is that since it is obviously isolated from bacteria which can survive in the very harsh condition, this Taq polymerase also remains active at the higher temperature during the denaturation of the double strand. So this is the reason why we use tag polymerase, because the 94 degree, because the 94 degree is needed for the separation. 94 degree is needed for the separation of the DNA strand. But then in the same, we cannot stop the reaction again, start and then again add the again add the enzyme, because every cycle it has to separate and then again you have to it has to the new stand again separation again new stand separation, everything. So it's a very continuous process. So so therefore the scientists were looking for a polymerase that was stable and active at 98 or 94 to 90 degrees, and that's why they got tag polymerase. Tag polymerase is a DNA polymerase isolated from thermal activities and is very stable at high temperature, such as 94 degrees, which is important for denaturation of the double-stranded DNA. So that is why we use tag polymerase. Now, through continuous replication, the DNA segment is amplified up to 1 billion copies. So you should remember this as well, that this cell-free method of amplification of the DNA of the gene of interest is very, very important, and it has pioneered the research in a lot of research in genetic engineering. So with the increase of cycle, there will be increase in the genome copies of the DNA. Now the amplified fragment can be used to ligate with the vector for further cloning. So from this we will get the gene of interest, and from there we can put it in a put it in a can put it in another vector, that is cloning, basically cloning. So now, so this is the overall view of the PCR, and we'll be ending up today's session with this thing, and I'll be summarizing of whatever we learned about this today. So this is basically PCR. This also known as not the machine is called thermal cycler, because it has this temperature thing. And so in a PCR reaction, what we want to put is, first we will put uh, DNA sample, and then to identify a specific gene, we'll put those primers that will be specific to it. Basically, it will bind here and here. So the primers. Then we'll put nucleotides, that is DNTPs, deoxyribonucleotides, the nucleotides, and then to extension. So this DNA tag polymerase uses DNTPs to amplify this segment. And then we'll have a buffer. Buffer is basically suitable for the enzyme for the better activity. And everything we'll put it in the PCR tube, and then we'll keep it on the thermal cycle. And then the PCR process takes place. The first denaturation, where the strands separate, it's at the high temperature. That a normal DNA will, after at 95 degrees, will separate. And in the handling step, which is at 55 degrees, the primers will bind on the separate strand, and they will amplify only this particular segment. And the extension is at 72. So you should remember the temperatures. It is 95, 55, and 72. And this is the whole process of PCR. So what we learned today is that we learned about agarose gel and of how it is able to separate DNA strands based on size. So this is what we learned about agarose. Then, in order to mass produce a gene of interest, mass produce, we have mass production. We have two options. One is cell-based amplification, and next is cell-free amplification. In the cell-based amplification, we basically transform bacteria, and then we culture, plasmid isolation, etc. And the drawbacks are, disadvantages basically, it is very tedious, because the larger the culture, the more hands we need to handle it, and more the chances of contamination, everything. It is costly because we use a lot of media, kits, and everything. Tedious, costly, and time consuming. So in order to remove these things, we follow a cell-free amplification method which utilizes PCR process, which is done in thermal cycler. So this PCR is basically polymerase chain reaction, right? This is done in thermal cycler. What are the components of PCR? If I want to do a PCR, what I need? I first need this thermal cycler, obviously, thermal cycler. Then in the master mix, that is in the PCR reaction, I'll need the DNA that is complete, then the primers, that are used to amplify my gene of interest. Then I'll need tag polymerase to amplify my gene. And to amplify my gene, there is extension. So for that, I'll need DTPs. And then for this tag polymerase, I'll need a buffer enzyme so that the activity of this tag polymerase can be enhanced. So these are the things that we need. Next, PCR has 3 steps. One is denaturation. Then we'll have annealing. Then we have extension. The denaturation we do at 95 degree. Annealing we do at 55 degree. And this we do at 72 degree. The denaturation we separate the DNA. In annealing, we anneal the primers. So, first step is to separate the DNA. Second step is to anneal the primers. And the third step is the elongation step. That is extension where the DNA will be here. And then after a particular number of cycles, it is mostly 30 cycles, we'll get a copy of our DNA. And if we run this on gel, we'll get only one separate clear band. We run on them, and that's why the PCR is very efficient process. And yeah, and this tool is being used up in a lot of things because it helps us to amplify the gene of interest very fast. Within 3 hours, we'll have a very intense and very high concentration of the DNA. So this is it for today from my end. Let's meet up in the next session, and we'll discuss more about this. We'll discuss more about this other process of the DNA of the of the biotechnology basically, and we'll finish up this session like this course in two more sessions, I guess. Okay, then thank you for joining. Let's meet in the next session. Thank you.